like Kelly said, my name is Matthew Deen. I worked at Simon Fraser for a while with Professor David Vocadlo, and currently I'm in Zurich working on peptide-based therapeutics for fibrosis. So a bit of a career shift, but excited to talk to you today about clinical trials and some of the current research that's ongoing towards Parkinson's disease therapeutics. So let me just see here to figure out how to change the slides. There we go, I'll just do that. So yeah, a little bit of a comic to begin. And as you guys read this, it really highlights the difficulty of identifying new therapies, and not only identifying new therapies, but demonstrating that they work. And that can be one of the largest hurdles that pharma companies or biotech companies
Hi, sorry, Matthew. I can't hear you anymore.
You can't hear me.
Yeah, before.
Okay. Just a second. Okay. Is this better?
Yes, I can hear you now.
Okay. I don't know what happened there, but okay, cool. So yeah, figuring out if something causes immortality is impossible, but figuring out if a therapy for Parkinson's disease or any human disorder is at the very least very challenging. So, and this is kind of where clinical trials come in, and clinical trials are just simply experiments that test new drugs or new therapies or a variety of different interventions in humans. And so what are clinical trials? They're just simply human experiments to demonstrate that a particular treatment or therapy is safe, is a big one, but then also that the intervention improves the lives of the people that use it. And what we're going to do today is we're going to go over kind of the framework for how this is accomplished together. So, but before we begin, we're going to do a little bit of a dive into the history of clinical trials and kind of see where we got to, or how we got to where we are today. And so the first kind of like foundation for clinical trials was established by James Lind in 1753. And what he did is he published the first comparative study for sailors that's suffering from scurvy. And so scurvy, back when you had long transatlantic voyages, was a hugely dangerous disease, and nobody knew what was causing it or how to cure it. And James Lind took 12 sailors suffering from the disease and divided them into pairs and gave each of the pairs different dietary changes. And so he tried things like vinegar, diluted sulfuric acid, and then the two sailors that he gave lemon juice to, or lemons, made a miraculous recovery. And so they didn't understand how the disease happened or what was causing the disease, but they found that lemons, and later now we know that it was the vitamin C in the lemons, cured scurvy. And that was kind of the foundation for like, how do you run and organize human experiments? Caleb Perry came in and looked at a new way in setting up clinical trials and looking at, and he did this by testing if British or Turkish rhubarb had different effects as laxatives. John Haygarth in 1799 was the first to use a placebo, and this was like a really big advancement in the area of human experiments. So a little bit of an explanation. John Haygarth was in an era where there was a lot of people that were selling metal rods as electromagnetic cures for a variety of different ailments. And John Haygarth was like, this can't be real. And so he made wooden equivalents that looked the same, and sold them. And he noticed that 4 out of 5 people actually reported an improvement with his wooden rods. And so disproves this whole electromagnetic metal rod theory that people were spending so much money on. And this highlights a point that humans have the ability to influence, that just the expectation of feeling better influences their mental state, and kind of their perception of how they're doing. And so a lot of clinical trials nowadays use these placebos, and we'll talk a little bit more about them later on. And then Claude Bernard really established the fact that you need to have comparative groups. So you have 2 patient populations, one that you benchmark as a control for the against the others. And so going on a little bit, just around the turn of the 20th century, Johns Fibberger described randomization as a key principle in designing human experiments. So as you can see, a lot of what we're doing is trying to eliminate human bias or our own ability to perceive things incorrectly, and in doing so, establish the most robust experimental results possible. James Byrne Amberson established double-blinded experiments, and so maybe you've heard of these, but you can have single-blinded and double-blinded experiments. So single-blinded experiments are if your clinician knows that you're getting a placebo or getting the active treatment. That's what we would call a single-blinded, but there's still room for intrinsic biases from clinicians towards placebo versus actual treatment patient populations. And then double-blinded experiments are where nobody knows who's getting the placebo and who's getting the active treatment until after the study is completely done. And so in doing so, you remove all human bias. And so that was a really key turning point. And then it wasn't until the 1944/48 in this Petulin and streptomycin study that we really established what we would call today the first clinical trial in kind of the era of modern drug discovery. And so it was a large multi-center placebo-controlled double-blinded experiment looking for treatments for tuberculosis, which is a huge problem for many years. So when we talk about clinical trials, a lot of the times our minds first go to therapeutics and drugs, and how they can, and the demonstration that they're effective, but a variety of different interventions can be studied in clinical trials. Because as I said, a lot of these clinical trials are just experiments using human patients. So you can test devices. And so an example is with Parkinson's disease patients, deep brain stimulation. Those went through clinical trials to demonstrate that there was a benefit to patients. You can have surgery to demonstrate that, to examine if surgery can help certain patients. So an example would be bariatric bypass surgery for those struggling with obesity, radiation therapy for cancer patients, diagnostic procedures. So you have to demonstrate that your ability to diagnose a certain patient is effective in a patient population. And then diet and lifestyle, also just simply investigating specific diets or lifestyle changes and the effects that they can have on disease progression is also an important thing. And so really what you can see is a variety of things can be tested, but today we're gonna focus on drugs and new therapeutics. And so kind of an overview with clinical trials and why are clinical trials done. So primarily they provide research data about whatever intervention is tested, and useful clinical data that's specifically used by regulatory bodies to decide what therapies or interventions might work. They also help to inform clinicians about what therapies and interventions might work for specific patient populations, depending upon where they are in the disease progression or what side effects they might be struggling with. Yeah, so we're talking about these big human trials and human clinical trials. We're going to take another quick little step back and talk about kind of where they fit in this landscape of drug discovery. And so there's three kind of main classifications of research. You have basic research, which is all very curiosity driven. And so this is stuff done at some of the academic institutions, a lot of academic institutions. So Simon Fraser and UBC are both fantastic institutions that do really great research. Then moving from that curiosity, kind of like fundamental science, you move into translational research, where you look to apply those discoveries that are made on a really basic level to human problems, and you try to investigate if you can apply them to human problems. And so there's an increasing push now for these academic institutions to be involved in this translational research to apply it and really help the general population. And then lastly, you have applied research, which is taking that translational kind of like proof of concept research and directly applying it to solving problems that people are currently struggling with. So the design of new medicines or devices. And it's this gap between translational research and applied research that's quite large, actually. And so it's at this point that academic institutions either have spin-off companies, and there's some really good examples from spin-offs from both SFU and UBC doing really nice work in the Lower Mainland, or these academic labs have to partner with larger biotech companies to move and really progress their research forward. And it's at this applied research level that you're looking to solve problems that humans are dealing with that you start to consider clinical trials. But as I mentioned, before you get to clinical trials, there's a lot of work that has to be done. And this is what we would determine as define as preclinical studies. So it involves identifying the target, your therapeutic target, so the protein or the biological mechanism that you wanted to manipulate with your therapeutic, and identifying a compound or a therapy that can do that. That's very challenging in and of itself. And so that's a main focus in preclinical research. And then once you have that, you want to apply it to different animal models to study initial indications that it works as you think it does. And so this can be done in a variety of different animal models, from mice up to non-human primates. And as is the case with all of clinical trials, a main focus is not only on efficacy, but it's also on safety. And so when you're working in these animal systems, you're monitoring toxicity, any potential side effects, really how this therapy that you're developing is influencing this animal that you're working with. And once you have indication that it might work and that it's safe, you have to develop a manufacturing protocol to demonstrate that you can do this reproducibly. And then after that, you also then develop clinical protocols, study plans to outline kind of how you will study this in humans. And then this whole package comes together and is submitted to Health Canada for an investigational new drug approval. And so it's at this point that Health Canada comes in and says, okay, cool, you have all of the background information, and you demonstrated that there's at least indication that it works and that it's safe. And Health Canada or the FDA in the States will come back and give you an approval to start testing your therapy in humans. And at this point, one of the things that's kind of important to keep in mind, and perhaps something that not most people have an appreciation for, is that it takes about 10 to 15 years to get this to this point, and about $1 billion. It's a very expensive and time-consuming process to develop therapies into something that goes into clinical trials. It's an exercise in patience in a lot of ways. So But moving on, so a lot of the times, not a lot of times, but sometimes, um, because you're working with humans, you're and we're different species, the way that we metabolize and distribute therapies throughout our bodies can be very different than the model systems and model animals that we work with. And so phase 0 trials, so even before you start your phase 1, 2, 3, etc., trials look to establish if what you saw in your animal models translates to humans. And so this is a really small study looking at where 10 to 20 healthy individuals are given really low doses of your therapy, and the way that your body metabolizes the therapies, and to make sure that the therapy reaches the correct part of the body, are all kind of main questions of phase zero clinical trials. And at this point, because you're using such a low dose, there's no investigation to see if there's a therapeutic intent. There's no therapeutic investigation. And if a compound looks good in phase 0 and it looks safe, then the compound is taken towards phase 1 clinical trials. In phase 1 clinical trials are still all focused primarily on safety, to make sure that there's no adverse effects, how much of the drug or therapy can you give to patients, and then what happens to the therapy inside of the body, where does it go, and what are the kind of decomposition products that your body produces when it starts to metabolize the therapy. And so this is a larger study, about usually 20 to 100 people, and sometimes you'll have both healthy and patient volunteers for these kind of studies. But because you're slowly increasing the amount of dose to try to find the maximum kind of tolerated dose or maximum effective dose, these clinical trials can sometimes be quite slow. So sometimes it's here that things go into phase 1 clinical trials, and it's a year or so before the results come back from that, and so it can be, again, a bit of a slow process. But if everything looks good and it's safe and there's no severe adverse effects in your population, it'll be taken forward to phase 2. But phase 1 is the first place in which we might see a placebo control. And so placebos are just inactive therapies that look like the active therapy, but don't have the active therapeutic compound inside. And so patients that are given placebo are just functional controls to demonstrate that if one patient population improves, it's due to the fact that they received that therapy. And so the placebos don't have any effect on the condition being studied. And so this can be in the form of sugar pills that look like the active therapies, or in the case of, let's say, the deep brain stimulation devices, when they were first going through clinical trials, it would just be certain patients wouldn't have their device turned on until after the study. And it's the primary goal of this is just to simply make the results of the studies more reliable, and a clearer proof that they are safe, or that they work as described. So yeah, after everything with this placebo-controlled phase 1 and everything looks good, clinical companies or biotech companies might move their therapy towards phase 2 clinical trials, and this is kind of really where the rubber meets the road when it comes to evaluating therapies, because it's the first time that people start to ask the question, does the therapy work? And that's kind of the point of phase 2 is an initial demonstration that these therapies work in humans. And it's done on kind of a medium-sized patient cohort of about 1 to 300 people. And it really is more of an initial investigation to see if it's worthwhile to test in larger patient populations in phase 3, because as you increase the number of people that join a study, the costs associated with it go up. And so phase 2 is more of like what we would call a proof of concept study in some cases. and other outcomes from phase 2 might be about the side effects and how to manage them. And it also is an investigation on what is the best dose, where do you see the most benefit to patients, what dose do you see the most benefit to patients at. And so, and this is all super important information for when you would move on to a phase 3 clinical trials. And so if you're looking through the news or if you're looking through new therapies, you might see phase 2 clinical trials being split into phase 2A and phase 2B. And there's a lot of these kind of different clinical trial names. So the phase 2A might be more preliminary and dosing and safety focused. Phase 2B would be on more extensive efficacy data and kind of like a mini phase 3 clinical trial. So it's at this point that it's important to talk about how one judges if something is working well, and the way that you do that is what we would call endpoints. And so within endpoints, you can have a bunch of different approaches to evaluating therapies. And so the first can be patient reported, so you could go through patient questionnaires. You could be clinician evaluated, and so this would be the MDS Unified Parkinson's Disease Rating Scale, which is just a doctor evaluating how a patient is doing in a multivariable kind of like setting. Imaging is also used within Parkinson's disease patients. So looking at amounts of dopaminergic neurons is something that correlates with disease progression and is used as an endpoint. And then within sometimes biofluid biomarkers, so blood samples, CNS fluid samples can be useful in evaluating how a therapy or how a patient is responding to a specific therapy and if it's having the intended effect. And lastly, because particularly for Parkinson's disease, because there is it's such a complicated disease to evaluate, and there's so much variability between day-to-day symptoms presented. There's a push to look at cell phone monitoring to make sure that you can track how people are doing, not only on like specific clinic visit days, but throughout their life, and really evaluating how a therapy influences the well-being of patients. And so I don't want to overload you with too much data, but this is an example of kind of how an endpoint, these endpoints are used. And so the primary endpoint, you'll sometimes hear that if you're reading through some of the news or looking through kind of reports on different therapies. The primary endpoint is the endpoint that's used to evaluate overall if the therapy is working. And then the secondary endpoints are just simply ones that are used to support that primary endpoint. And so this is an example. AstraZeneca is evaluating exenatide in phase 3 clinical trials for Parkinson's disease. And the way that they're doing that is they're using that clinical evaluation, the MDS UPDRS, the one that we mentioned in the slide previous, to evaluate how this therapy is helping patients. And then their secondary points are a lot of self-reported kind of day-to-day experiences of patients, and then a few other clinical evaluations. And this is one of the important things is sometimes they'll say, oh, you'll hear like, oh, this therapy didn't meet its primary endpoint. But that doesn't mean that it didn't help in some of the secondary endpoints. And so sometimes it's useful to go back, and sometimes companies go back and they go, oh, it didn't function exactly as expected, but there might still be some benefit to patients because we see this evidence in our secondary endpoints. And so, yeah, so let's say everything works well and you go to phase 3 clinical trials. This is really a demonstration on a large patient population that your therapy works. And so use 3,000 to 3,000 people, and this is usually longer term. usually, and for Parkinson's patients, usually 18 to 24 months. And it's a good demonstration that the therapy works. But you also collected data on what the side effects are on like a chronic setting, and then potentially just rarer side effects, which are all super important to know, to make sure that a therapy is particularly safe and for people to take in a larger setting. And yes, because one of the other main points in phase 3 is how does the treatment affect the people's quality of life? And so let's say everything works out and phase 3 clinical trials go great, and you see a huge benefit to patients. It's at this point that you collect all of your data and you submit it to Health Canada for approval, so that you can bring your therapy to the general population. And it's at this point that your therapy would be approved or denied approval to be sold based upon the data that you've collected. And this is evaluated on the safety and the efficacy and the benefits to patients. So, and then after the approval, sometimes you'll hear phase 4 clinical trials. And this is kind of after things have been licensed to be sold to the general population. They'll continue to monitor the effects of this therapy to make sure that it's both safe and effective to like a much larger population. And then, and this is kind of like the last step for the clinical trials. So kind of an overview or a summary of clinical trials and kind of the structure, and really a broad view of it is the preclinical phases that we saw with the studies in the lab and the animal models. You can think of that as like a developmental phase. So you're developing the therapy. And then the phase 0, phase 1, and phase 2 are really exploratory trials with humans, where you're like you're trying to learn about the safety and understand how your therapy works, or if it works, as is in the case of phase 2. And then phase 3 and phase 4 are what I would call confirmatory phases. So they confirm both the safety and the efficacy of your therapy. And so kind of an overviewed structure is the standard approval method, kind of mechanism, is that you have your discovery and preclinical phase 1, phase 2, phase 3, and then it goes to the review by Health Canada or the FDA, and then it's either approved or rejected. But one of the things that you might see with Parkinson's disease, Parkinson's therapies, or other therapies in general, is alternative structuring of approval pathways, specifically for the FDA. And so you have something called accelerated approval, which is you have phase 1 and then phase 2 and 3, almost a shortened phase 2 and 3 that uses instead of the clinical endpoints, kind of surrogate endpoints, so easier to measure endpoints. And then after those perhaps more simple phase 2 and 3 clinical trials, it goes out to FDA review. And then if it's approved, then you have a phase 4 clinical trial to demonstrate in a larger patient population that it works. And you can achieve you can receive the accelerated approval for your therapy if it is a drug that treats a serious or life-threatening condition and fills an unmet medical need. And so this is something that Parkinson's disease therapies are currently, a few of them at least, are being evaluated under. And then the fast track is another one where you have just simply phase 1 and phase 2. You have an initial indication that it would work, and then you have FDA approval, and then potentially into a phase 4. And this structure is just used for therapies that just might take longer to demonstrate their efficacy, their effectiveness in treating patients. And so you've demonstrated that it's safe, but you might need more time to demonstrate that it works. And so this is another structure that you might see when you start to think about and read some of the kind of reports on the development of new therapies. And so we've talked about clinical trials, and there's a few kind of perspective points that I want to bring into this, and that is how frequently does a therapy get approved, and how many therapies enter clinical trials versus how many therapies are actually approved. And so what you can see here is on the left is a diagram that outlines the success rate of therapies over a historical success rate of therapies as a function of different phases. So in phase 1, you can see we have about a 50% success rate, 60% success rate nowadays, just pretty high. And then in phase 2, it's closer to 35-40%, and so it really is that demonstration of efficacy that can be a main hurdle for a lot of different companies. And then phase 3 again is back up to that 65-70, but the overall success rate is about 10%, and this really depends. I have to put in the caveat that this depends upon the disease and the disease type that these therapies are targeting. So neurology is one of the ones that is closer to 8 or 7% versus other fields that have higher success rates, and it just highlights the difficulty of targeting brain-based diseases. It really is something that's not simple, and something that scientists are still really struggling with. And then the other thing that's important to highlight is the cost of developing drugs. So these are maybe a little bit dated numbers, but as I mentioned before, you can expect to spend about $1 billion before a drug even enters clinical trials. And then during clinical trials, you can expect to spend at least that same amount. So the current estimates are about $5 billion per therapy developed. And this really highlights the percentage, the cost of developing therapies as a function of revenue. So pharmaceuticals and biotech are one of the highest R&D, research and development spending companies, because it just is simply difficult to identify and kind of figure out how to treat diseases. Humans are just inherently very complex. So moving on, I wanted to talk about some of the examples of things that I think are up and coming for Parkinson's disease patients, and I wanted to highlight a few kind of interesting and exciting treatment methods that people are investigating. And so here on the right is a figure that highlights and that demonstrates all of the different therapies that are currently in clinical trials. So you have disease-modifying therapies on the top half of the circle, and then symptomatic treatments on the bottom half of the circle. And because Parkinson's disease is such a complex and poorly understood disease, many different approaches are being taken to try to find something that really helps patients, and there still is that desire and really drive to identify disease-modifying therapies, because there hasn't been one yet. And you can see that there's many in phase 1 and a bunch in phase 2, and then once you get to phase 3, there's a few here that we'll highlight. And so, like I said, exenatide is in there, and so it'll be interesting. I think that the clinical trials for exenatide is coming up to a close soon, so that'll be something to keep an eye out for. And the first thing that I wanted to talk about was neurodegeneration targeting antibodies. And so, like I said, with Parkinson's disease, the exact cause and driver of the disease is complicated, and we're not exactly sure how it works. But one of the hallmarks of the disease is the development of different plaques within the brain. And here we have these alpha-synuclein aggregates. And one of the approaches that people and pharma companies are taking is to develop antibodies that bind to these plaques, because it's thought that these plaques are the cause of some of the loss of brain function. and these antibodies bind to the plaques and then result in them being broken apart and alleviating that kind of that buildup of toxic proteins within the brains, and hopefully leading to an improvement in patient health. And so kind of a success story here, not for Parkinson's disease, but for Alzheimer's patients. Leqembi, which was brought to the market by Biogen, was just approved, and it targets not the alpha-synuclein aggregates, but tau aggregates in Alzheimer's patients, and it showed a 27% slowing in cognitive decline, as judged by that clinical evaluation that we talked about. It has to be said though that there can be some severe but transient side effects, such as dizziness, nausea, and seizures that can be associated with this treatment. But those tend to go away. But this is kind of the first example of a disease-modifying therapy for Alzheimer's, and so this approach has been taken as well for Parkinson's disease treatment. And so currently there's this antibody that targets these alpha-synuclein aggregates, prasinezumab, which binds those alpha-synuclein aggregates, and there was a phase 2 clinical trial that was reported, which showed no overall effect in the primary endpoint, like we talked about, but they really noted an effect in two of their secondary endpoints, and so they're fairly optimistic that this might have some disease modifying and disease slowing capacity. They're just working out how to evaluate that more effectively. So they're doing a longer term phase 2b clinical trial to investigate the effects of this antibody on specifically on motor symptoms. And then kind of a really interesting approach that some companies are taking is not a passive treatment with these anti-alpha-synuclein antibodies, but an immunization, a vaccine against Parkinson's. So developing therapies in which you prime your own immune response to target these alpha-synuclein aggregates and degrade them before they start to accumulate or after they started to accumulate. And so company Vaccinity has just started phase 2 clinical trials with their Parkinson's disease vaccine. And they had some initial results from their phase 1 clinical trials that suggested that there might be some efficacy in treating Parkinson's disease patients. AC Immune also is starting phase 1/2 clinical trials to evaluate both tolerability and efficacy in this Parkinson's disease vaccine. And so, like I said before, Parkinson's disease is a complicated disease, and one of the main limitations that we have is we don't fully understand how it works and what causes it. But we do know that there's certain proteins and genes that are associated with familial or sporadic Parkinson's disease. And these are shown in this figure here. And so there's been a lot of effort in the past 15 years or so to try to target these specific proteins to identify new Parkinson's disease therapies. And so when on a little bit of a side note, just want to highlight some of the work that the Vocadlo group has done with the generous support of both the Michael J. Fox Foundation and Parkinson's VC. So Professor David Vocadlo, when I joined the lab, he was very interested in this protein GBA, or this gene GBA. And what has been found is that in patients with Parkinson's disease, there's less of this protein and it's less functional. And so we wanted to develop a system that we could use to study this particular protein within the cells of patients or model cells. And so what we did is we developed a system that we could administer this tool to cells, and they would take this dark substrate, we call it a dark substrate, and then GBA would turn it into something that would become fluorescent or bright, and we could visualize that. And so we could directly see the function of this protein within cells, and we could measure the amount of this protein that was there, which is something that nobody had done yet. And so we wanted to see, could we use this to identify new therapies that would increase the amount of this protein function, and hopefully in doing so, eventually help treat Parkinson's disease patients. And so what we did is we would take Parkinson's disease cells, or a cell model. We would treat them with a different therapy, different therapeutic small molecules. We would use this new tool that would go from that dark to that light state, to measure how much of this GBA protein was functional inside of the cells. And we could measure that using this microscope, and we would get these nice images, which we could use to quantify the amount of this GBA function. And so we hope to find small molecules that increase the amount of this enzyme or this protein and how much it functioned. And those molecules could serve the foundation for new therapies. And so this was really a team effort from people within the lab to develop this and apply this. And so once we had everything established, we worked with the Center for High Throughput Chemical Biology at Simon Fraser, which is really a world-class research hub that has a robotics platform, so we can evaluate and test many compounds at once to rapidly find something that might function, that might serve as the foundation for therapy. And so we tested 65,000 different potential therapeutics, over 65,000 potential different therapeutics. And we found a few interesting compounds, and those are currently being evaluated further, not by myself, but other people within the Vocadlo lab. And as Kelly mentioned, we're also working to apply this that has been, as I said before, supported by both the Michael J. Fox Foundation and the Parkinson Society of BC, to other translational applications, such as a clinical assay to support those different endpoints in clinical trials. So we're not the only ones that are interested in GBA and GBA targeted therapeutics. There's been a bunch of other different groups. And so there's really a sense of hope that these compounds that target GBA might serve as new therapeutics. And so this company, BL, has actually identified one and has developed it quite far. And it's currently in phase 2 clinical trials. And those are currently actually ongoing in Canada, which is quite exciting. And they were just initiated last year, and so it'll still be a year or so before we find out the results of that phase 2 clinical trial. And so using this tool that we've generated, we've also partnered with Denali Therapeutics, and Denali Therapeutics is interested in this other enzyme called LRRK2. In LRRK2, it's not the reduced function, but actually the increased function of this enzyme. And that actually causes a reduction in GBA. So there's some interesting things there. But they've developed this compound, DNL151, and it inhibits, so it reduces the function of this LRRK2 enzyme. And that's currently in phase 2B clinical trial, so it's being evaluated for efficacy currently, and that's coming up. I think those clinical trials are ending fairly soon. But I think that this approach of targeting these genetic risk factors is quite exciting, and something that hopefully will lead to the first disease-modifying treatment for Parkinson's disease. If you're interested, that's all of the therapies that I'm going to cover for now. If you're interested in reading more about what therapies are being evaluated and kind of where they are in clinical trials, and getting up-to-date information on how those are progressing. There's this website, allsforum.org/therapeutics, which can be super useful. It highlights all of the Parkinson's disease associated therapies that are currently being evaluated, and gives a really nice, concise summary of what the results have been, what people have seen, what the clinicians have seen with these compounds, and kind of how it's looking going forward. And then if you're interested in reading about what clinical trials are currently ongoing in Canada, there's actually a database of all of the clinical trials. And if you look up specifically Parkinson's disease clinical trials, you can find all of the ongoing Parkinson's disease clinical trials in Canada, and understand kind of maybe some of the therapies and therapeutic approaches that are being taken. But yeah, that's all that I have for today. I'd like to open the floor to any questions that people have. I'd be more than happy to try to answer them. But before we go there, I guess I'd like to thank the research group that I was a part of in doing some of the work that I briefly showed you. David Vocadlo really is a standout human, and does some really fantastic work on Parkinson's disease. And I'd like to acknowledge the sources of funding for this project. So the Parkinson's Society of British Columbia was very generous in supporting this work. The Michael J. Fox Foundation has been very generous, and then Glycomed and NSERC, two Canadian research organizations that have supported this work, and we're thankful to both of them. But yeah, so for now, if there's any questions, I would love to try to answer them.